In our first chapter in the STEM Challenge is going to be Chapter 3, which is an introduction to the cells, Section 3.1. We're going to be looking at the cell theory, we're going to be looking at the diversity of cells, we're going to be looking what all cells have in common, but also the difference between prokaryotic and eukaryotic cells. But first, before we get to cells, we need to go back a little bit and talk about the elements. Take a look at this periodic chart. Most of you are familiar with the periodic chart, and the periodic chart tells us everything in the world, really in the universe, that's made of matter. Anything that's matter, anything that we can touch, feel, weigh, see, is made of matter. It's a combination, one or a combination of these elements. For example, all life is made of primarily these four elements: carbon, hydrogen, oxygen, and nitrogen. Now, there are other elements that also take part in life, like calcium and sodium and potassium, but the main four elements, just remember CHAN, carbon, hydrogen, oxygen, and nitrogen. Those four elements form the four main molecules that make up most living things. Now remember, these four molecules, and we tie them to an important word so you can remember what they do. Proteins are the workhorses of life. Carbohydrates are the energy molecules of life. Lipids are when we store that energy. When we need to store that energy, we store it in lipids. Another name for lipids are fats. And some of the most famous molecules are DNA and RNA, and those are the information molecules of life. So remember, those four elements, carbon, hydrogen, oxygen, nitrogen, combine to make these proteins, these carbohydrates, lipids, and nucleic acids. Sometimes students ask, why do we call them cells? Well, the name cell came from Robert Hooke. Robert Hooke created a number of different microscopes to see things closer. And one of the first things he looked at was a piece of cork under his microscope. And when he looked at cork is a plant, he noticed that there were all these little compartments. Robert Hooke said they looked like cells in a prison, and that's where we get the name cells from. Remember, it's important in our STEM Challenge course that you not only listen to these screencasts, but it's also really important that you read the chapter of the book and take notes, because we're just going to highlight some of these, but you should read about Hooke, and then you'll also be reading about Anton von Leeuwenhoek and what he did for microscopy. By the early 1800s, a lot of scientists were using microscopes to study different organisms, and that led two German scientists by the name of Theodor Schwann and Matthias Schleiden to propose that cells were the basic building blocks of all life. And then a German doctor, Rudolf Virchow, also contributed to what now is known as the cell theory. So these three scientists, Schleiden, Schwann, and Virchow, led to the cell theory. And these are the three tenets of the cell theory. All organisms are made up of one or more cells. All life functions of organisms occur within cells. And all cells come from existing cells. And that's pretty amazing when you think about it, because you think of the one cell that you started out as came from your parents. The one cell that they started out with came from their parents, and back and back and back generations. All cells come from pre-existing cells, and these three tenets are known as the cell theory. Sometimes students wonder why cells are so small. Most cells are tiny, and we're going to talk about the size in a few minutes, but the reason they're tiny is because lots of materials have to move into a cell quickly and efficiently, and lots of materials have to move out of a cell. And that's why the surface area to volume ratio is so important. As cells get bigger, there's a tendency for them to become more inefficient. So this diagram here in 3.4, where it talks about the surface area to volume comparison, is showing you that a small cube, a small cell, will be much more efficient than a larger one. Cells come in wildly different shapes and sizes. Take a look at these. This is a nerve cell. These are blood cells. This is an electron shot of bacteria. Here are algae, and here are pollen grains. So look how different they are. And the shape of a cell is tied to its function. For example, this nerve cell needs to carry electrical signals called impulses, and we'll talk about them in a chapter later on in the course. But the important thing for this page is all different types of cells are different shapes and sizes due to their function. So let's talk about cell sizes and how big cells are. But first, you have to know that, for example, 1 millimeter is one of the smallest marks, gradations on a ruler that we use regularly. And a millimeter is still way too big to measure cells. So what we do is we take 1 millimeter, we divide it into 1,000 pieces, we call them microns, and the abbreviation for micron is a Greek So for this course, one of the things we want you to do is be familiar with microns. So for example, most bacteria, or prokaryotic cells, are between 1 and 10 microns. They are really, really tiny. Most eukaryotic cells, we'll talk about them later, the cells that we're made of, are from 10 to 100 microns. So look at that 100 microns. So 100 microns is 1/10 the size of 100 microns. Down here, for reference, we have the diameter of a human hair is around 20 microns. Most of the slides you see for our course will be from our textbook, but occasionally I'll find a slide or a picture that I want to use or a video that I'd like to use, and the last 2 slides are one of those. So you can always come back to this and take a look at this slide here. So this is visualizing slides. So a human being, if we wanted to measure a human being, we could do that with meters. If we wanted to measure an egg of a hen, that would be in centimeters. But once we get past millimeters, there's 1 millimeter, we need to use microns. So a human egg might be about 100 microns. A red blood cell might be 10 microns. Notice that our light microscope can see these dimensions. But once we get down to things like viruses or molecules, we can't even use microns anymore. We have to use 1 micron divided by 1,000, which are called nanometers. We won't talk too much about nanometers in this course, but we are definitely going to be talking about microns and the size of cells. Although cells are really different from one another in different shapes and sizes, they all have some very common parts. And these might be the plasma membrane, or you'll hear me call it the cell membrane. So plasma or cell membrane means the same thing. The cytoplasm, the liquid that's inside the cell, ribosomes, all cells have ribosomes, and all cells have DNA. So they're listed here for you. They talk about the plasma membrane or cell membrane, cytoplasm, ribosomes, and DNA. Here there are 2 general types of cells. There are prokaryotic cells, and these are cells that don't have a nucleus. Like if you look down here, this is all bundled up DNA, but there's no central nucleus. The DNA, as they say in prokaryotic cells, is in the cytoplasm. These typically are compared to bacteria. So whenever you hear the term prokaryotic cells, think of very, very small bacteria. One of the things that we do with our course is we'll also be reading some of Ed Young, and you'll become very familiar with prokaryotic cells through reading chapters in that book. Now we're going to go to a cell which is much bigger, the cells that we're made of, the cells that do have a nucleus, and those are called eukaryotic cells. Although you can't see it from the previous picture, the eukaryotic cell is much, much bigger. Let me give you an example. You see this right down here. This is a mitochondria. That's about the size of our bacteria. So you can see that this is a much, much bigger cell. And eukaryotic cells, we're going to compare and contrast them throughout the course, but I'd like to point out a couple parts here. So on the outside here, we have the cell membrane or the plasma membrane. We have the nucleus, which is over here. We have Golgi apparatus, we have mitochondrion, we have ribosomes. So we'll mention these parts, and we'll tell you a little bit later what parts are the most important to know. So let's briefly review in this lesson summary what we've talked about in section 3.1. We talked about that it was the microscope that led to the development of the cell theory, that Schleiden and Schwann saw all those cells are basically the basic part of all life. Cells are super small because they have to have materials come in and out. That's the second bullet point. Cells are very diverse, and by diverse they mean all different shapes and sizes, but they all contain a plasma membrane or a cell membrane, cytoplasm, ribosomes, and DNA. Prokaryotic cells, those are the small ones, are similar to bacteria, are cells without a nucleus. And eukaryotic cells are larger, and they have a nucleus and other organelles. Organelles are like tiny organs. So that ends section 3.1.